We'll transition over now to Gordan Lauc. I don't know if I'm saying that right, from GlycanAge, who I've seen present before, and really cool company also. So take it away, Gordan.
Hi, do you see my screen?
Yes.
Okay, perfect. So I'm Gordan Lauc. I'm the Chief Science Officer of the company GlycanAge. And I will try to tell you something about how we can use glycans as a tool to prevent age-related diseases. So first, what are glycans? Not so many people know much about glycans. Glycans are the ultimate layer of molecular complexity. So practically all proteins which function on the surface of a cell or outside of a cell are covered with this furry glycan coat. And these glycans are not only a decoration, they're actually playing active part in interaction with the receptors, and this is as glycoprotein, and this interaction of the S glycoprotein with its receptors. And majority of proteins are glycosylated. So actually, to understand biology, we have to look into glycans. Unfortunately, many people still ignore them. And even the director of NIH at the beginning of this pandemic was showing the S glycoprotein without even a single glycan. And before I go on, I have to warn you that glycosylation and glycans are something completely different from the glycation, from HbA1c, from AGE. So these are just different things. Please do not extrapolate your knowledge about any of these things to protein glycosylation and glycans I will be talking about. So the GlycanAge is a London-based startup which already has a product on the market, a home test kit called the GlycanAge, which can be shipped globally, and it's actually on the market for over a year now. And it's based on a research done in my other company called Genos. Genos is a private research institute which currently has over 50 people employed, and there we got over 15 million euros of different EU, American, and national grants, which enabled us to study over actually 150,000 people so far, and not any 150,000 people. So we collaborate with some of the best researchers in the leading institutions like Harvard, Hopkins, Yale, Oxford, Cambridge, and we add glycans to all other data they have in their cohorts. So when we add all these things together, when we do a multi-omic analysis, then we can try to understand what glycans are actually doing and what glycans can prevent or can predict, which enabled us to publish really many papers. I think we have nearly 200 papers so far, around 100 of them are the basis of the GlycanAge test of biological age. And some of these papers are in the leading journals, including a couple of Nature group journals, Diabetes Care, when we talk about diabetes, gastroenterology for inflammatory diseases, circulation research, and so on. So one of the strong points of the GlycanAge test, there's really hard science behind it. And another good thing about the GlycanAge test is that it is kind of filling the gap of what is missing in other tests of aging, because majority of other tests of aging are good in many aspects, but they're lacking one of the key elements, and this is the responsiveness to the intervention, which is known to be beneficial for the biology of aging. To the contrary, we have shown in a number of studies that glycans actually respond to this type of intervention, and that glycans change when you do something good for your health. And another key aspect of GlycanAge test is that the molecules which we measure, glycans attached to immunoglobulins, are the functional effectors of inflammation. They're balancing inflammation, they're active participants in the process called inflammageing, meaning how inflammation is promoting aging. And if we remember the cover story from the New Scientist a little bit more than a year ago, you are only as young as your immune system. And we have to think about the immune system. We have to reset our immune age. And here, IgG glycans are the functional effectors which actually do the work. So if the glycan clock is improving, this means we have more of the good molecules. and less of the bad molecules. And the age of the immune system became so, the importance of the age of the immune system became so visible in this pandemics, where even in the early stage, and this is data from March 2020, the mortality is in the old population. So this virus was killing old and frail people, and subsequently we learned that in a younger population, majority of people which actually died were not metabolically healthy. So this is something which David Sinclair and I predicted in March 2020 when we said that the biomarkers of biological age, and we specifically mentioned the GlycanAge then, would actually be a better predictor of the severity of COVID-19 than just age itself. And glycans are here very good because they integrate genetic information, they integrate epigenetic information, they integrate direct environmental factors like stress, smoking, diet, exercise, and so on, into a simple index of age that can be reliably quantified. So these are molecules which you can take off the immunoglobulins, you can measure them, and provide reliable readout. And we actually later managed to demonstrate that this is in fact the case, that actually severe COVID-19 associates with older IgG glycome. And we are just now drafting a paper showing that actually people who had a more severe COVID were undergoing much faster changes in this age-related glycans than people with a mild or asymptomatic COVID. But COVID is just one of the examples. We studied dozens of diseases, and in most of them, we see that people with the disease have changes which are in the same direction as aging. So these are the effects of aging in different cohorts, and these are the effects of different diseases. So young person with a disease looks more similar to an older person than the person of its age that is healthy. And here again, I have to remind you that these IgG glycans are functional effectors. And what we see here very often is that glycans change before disease actually gets diagnosed. And the kind of a big picture is that maybe by using this type of biomarkers like a GlycanAge, we can identify people on a path to the disease, because normally people ask for help when they already have a chronic disease, when there is a pain, when something does not function, and this is beyond the point of no return. While if we catch the changes here, then maybe a simple lifestyle or preventive pharmacological intervention can restore the homeostasis, and then people can delay or even completely prevent disease development. And one of the good examples where we have a lot of data are cardiovascular diseases. So our first study on slightly over 3,500 people showed that the composition of the IgG glycome, so depending which glycans you have at a given moment, associates with the cardiovascular disease risk score. So other ways how to measure cardiovascular disease risk. And then using this data, we went to the Cohort. Cohort, the German part of Cohort collected 27,000 people nearly 30 years ago. They have samples from 30 years ago with the clinical follow-up. So we were able to see whether glycans can predict actually future cardiovascular event, heart attack and stroke. And for example, in women, we showed that a single glycan plus age is more predictive than the entire AHA score. And this was published last year in Diabetes Care. And also, subsequently, using a cohort of twins, which was sampled 3 times over 15 years, 2,000 of them, we showed that the glycans predict future hypertension, which was subsequently validated in another cohort from Munich with Christian Giger. And the most exciting part is that, at least in animal model, this can be prevented. So immunoglobulin glycans seem to be causing hypertension, and when they lose sialic acid, which happens, for example, if you feed mice with a high-fat diet, they become obese, they lose sialylation of IgG, they develop these old glycans, and these glycans bind to the receptors endothelial cells and promote hypertension. But if you feed mice with a dietary supplement called MANAC, which is N-acetylmanosamine, which is a precursor of sialic acid, these mice do become obese on high-fat diet, but they do not lose sialylation. So the sialylation is saved by this supplement, the dietary supplement. And despite becoming obese, these mice do not develop hypertension. So mice fed with a dietary supplement which promotes IgG cellulation are actually saved from hypertension development in the future. Another interesting example is when we look in I forgot to mention here, this is mouse data. We don't have a human data for this thing yet. What we see in humans is that, for example, in women approaching menopause, there is increase in this change of biological age. So while men are blue here, change more or less linearly in the entire age spectrum, women have this drastic change around the menopause period. So for example, what we are seeing with the GlycanAge clients is that somebody can have very good GlycanAge for years, and then around the perimenopause period, it deteriorates rapidly. And actually this change happens before any other symptoms of menopause are visible. And maybe this change in glycans could be one of the precipitating factor related to all the menopause diseases which appear in the menopause. So the key question we all have is, can we revert the GlycanAge clock of biological age? By reverting this clock, we delay or prevent age-related diseases? Because again, these molecules are not only biomarkers, they're functional effectors which balance inflammation. So if we change this type of glycans into this type of glycans, we know we can prevent hypertension, at least in mice, but maybe we can also prevent many other inflammation-related processes. And there is a lot of anecdotal data with individual patients. For example, with the Joseph Raphael in New York, he has over 150 people already tested for GlycanAge. We regularly see the pattern of people entering the intervention and then becoming significantly younger in a couple of months. We see similar things on this side of Atlantic. For example, Tim Spector is a famous genetic epidemiologist. We collaborate a lot with him, and we started to track his GlycanAge over 5 years ago. And he was very good for a long time, and then he rapidly deteriorated, and then decided to do a lifestyle change. And if you're interested what he did, there is an Instagram live about it. And then he improved his GlycanAge for 11 years in 8 months. So these things are doable, but this is still anecdotal. This is not science. So we are trying to do hard science by looking for cohort with the proper placebo-controlled groups. And this is one of the studies for the hormone intervention. So there was a group of women where the menopause was chemically induced, and then in a placebo group, they did not receive anything. In intervention group, the estrogen was restored with estrogen patch, and the placebo group aged on average 9 years within 6 months. Estrogen group did not. So if you take away estrogen, GlycanAge goes up. After recovery, they return to the normal values. And actually, women who had a better GlycanAge at the beginning changed the most, while the woman who had the worst GlycanAge did not change that much. Similar thing for men. If you remove testosterone, they increase GlycanAge. If you return testosterone, it does not happen. But if you block aromatase, which prevents conversion of testosterone to estrogen, it does not help. So it's actually estrogen which is regulating GlycanAge in men too. Another thing which definitely works is the weight loss. Bariatric surgery is very effective in rejuvenating the IgG glycome. We had even a patient that improved for 37 glycan years in 6 months. Bariatric surgery is maybe a little bit drastic for many people, but even a regular weight loss works. We were tracking the 2000 twins I mentioned before for 15 years, and twins who were gaining weight were aging faster than twins who were losing weight. But there is no magic diet. We tested 1,000 people on 5 different diets, and on each of the diets, including the high glycemic index diet, some people got worse, some people improved. So there is no magic diet. Weight loss works for everybody, but the optimal diet has to be found individually. The same goes for exercise. Many people believe they can sweat out bad diet. This does not work. You cannot sweat out junk food that you eat. And many people who were overtrained actually got worse. And there has to be an optimum for good results. And we have seen probably that this high intensity interval training works the best. Microbiome is extremely important. We have a number of studies linking microbiome to the glycan. For example, fecal microbiota transplantation has a strong effects on a glycan, and I don't have much time to go into details. So there is a very good review published by these two ladies from my group. It's freely available on this address here with over 500 references, everything we know about the IgG glycan today. So the idea behind GlycanAge is that we can actually help and motivate people to live healthier, because most of us knows what is healthy and what is unhealthy, but this is too difficult, and we don't make these difficult decisions if the reward will come after 20, 30 years. So we can always postpone it for later. But if we have objective insight today, if we see effects in a few months, if we have this tool which can quantitate interventions, then people get more motivated. And this GlycanAge is available. It's a global web shop, also available with a number of clinical partners all around the world. It can be ordered as a sample collection set, just a simple dried blood stain shipped back in the lab, and then you get results. And as I mentioned, this is all backed by hard science. We have over 20 million euros in different grants that we got in the last 10 years. And I have to acknowledge their generous support, which enable us to do everything what we did. And I thank you all for your attention.
Sonia, if you can come on.
I keep trying to come on and my video is not letting me. I'm here now. That was a great presentation. I am so motivated to eat healthy today now. Yeah, I love this. You have such great data. How far are you? You mentioned that you don't have any human suggestions yet for, like, you know, what you should eat when your glycans are not going the right way. I mean, how far are you from having like a dashboard where I can just hook up and it tells me, hey, you know, you need to get more selenium in your diet or something like that?
So when I said that we don't have a data, I said this is for a MANAC, which is a dietary supplement. We have a lot of intervention data on people, and I think the only conclusion is there is no standard human. So I think the good diverse diet is good for everybody. So supplements generally help people who are lacking specific things. So if you are lacking something, then the supplement can help. But in many cases, supplements don't help. So it's just the balance of healthy lifestyle, healthy diet, moderate exercise, not too much stress, which is kind of general advice. But the idea of this test is that you can actually see what is the most important for you, because for some people, it is stress. So you can even eat a little bit more, but if you control stress, you go in a good direction. For other people, they can cope with stress, but the diet is the problem. So I think this tool, this test, gives you a way to see what actually works for you.
And would I do it every week, or like would somebody do this every month, or how often would you do it?
So if somebody has a good GlycanAge, I would say once a year is more than enough. If there is an intervention, we would expect to see an effect in a couple of months. This is a very long-term measure. So if you change something, you will see effects in a few months.
Got it. Okay. So you have to be patient.
You have to be patient. And for example, well, I own the company, so I test myself every month or 2 months. But normally people, they do intervention and we tell them, let's try it after 3 months. Got it.
Got it. Now, somebody has put a question in the Q&A here, so I'm going to ask that. It's from Claude Lardanos, and he asks, I believe glycans play an important role in glycocalyx. Is that correct?
Glycans are the glycocalyx. So one type of glycans form the glycocalyx. These are the long polysaccharides, and they actually, this is one of the key ways how we communicate with our environment. For example, we feed the bacteria we like with our glycocalyx.
And so another question from me. So this is a biomarker, obviously, for aging. Could this also be used to like test the efficacy of drugs and things? So if you've got, you know, so say we're using in the future, one of Shift Bioscience's molecules that impact genes, and we want to see if the rejuvenation is actually happening. Could we use your test as a biomarker?
So it works on a level of an organism. You cannot test it on a cell system. We do have expression system where we look for genes which regulate IgG glycosylation. So there we can also test it on a molecular level. For example, one of the things we focus a lot is the menopause and estrogen. So hormone replacement is something which we can easily monitor with the GlycanAge and see whether it works or not. So we think that the GlycanAge is a kind of HbA1c for estrogen, because the problems in the perimenopause is that estrogen goes up and down, and you don't see the long-term effects. While the glycans will tell you the average estrogen concentration in the last few weeks.
Yeah, that was interesting that the men are just as impacted by estrogen rather than the testosterone.
Yes.
Interesting finding there.
Yeah.
I think that's a great place to focus your business, by the way. It sounds like you're going to focus there with the perimenopause and basically FemTech.
This is something we are also doing, yeah, and we are also developing a proper diagnostic test which would go into the routine clinical use. It will take some time because there are no validated machines and the chemicals, but we are moving in that direction. Yeah.
Great. Well, I think we should probably move. Thank you so much for that, Gordan.